So first is Matthew Miller. So Matt is the second from my left. So Matt is an associate professor of biochemistry and biomedical sciences at McMaster, as you know. He's also a molecular virologist working primarily in the area of influenza, but now, of course, on COVID. And you've seen Matt probably many, many times if you've turned on the news at any point in the last 2 years. He's been out there making sure that we get the proper information out there here, on COVID in particular. And he's been one of the main leaders in the development of our inhaled vaccine that you probably have seen and I mentioned earlier today. So Matt got his undergraduate degree at Western University, and then did a no, is that right?
Mm-hmm.
And your PhD in the same place. So see, I should never have read this. Now I'm looking foolish. But importantly, he went for his postdoctoral work, he went to work with Peter Palazzi's lab at Mount Sinai in New York City. One of the world's most famous and influential researchers in virology and influenza in particular, where he's been developing technology that he's brought back here and ideas about how to develop a universal flu vaccine so that we're not constantly chasing after the different variants that are coming out, so you don't have to go to Shoppers Drug Mart every year to get a vaccine, hopefully. He's he's a CHR new investigator, got the Baggy Singh Early Career Award, Government Ontario, from the Government Ontario. It's been cut off. Anyways, he's really famous. Right? And he's an absolute gem, and you're going to love what he has to say. So I'm going to scrap these notes. Amy Gillgrass is an assistant professor at McMaster University, one of our new captures, one of our new hires. And Amy's niche is the wrong word, but her amazing thing is to develop humanized mice, so mice with a human immune system, as you'll hear, so that you can actually study human-only viruses and infectious diseases. And in particular, what she has developed is a co-infection model of HIV and tuberculosis, which, if you know anything about challenges in the developing world, especially in sub-Saharan Africa, it's a major, major burden. So having a model for us to be able to study this is a really important thing, and so that's Amy's thing. So when we talk about level 3 facilities and all those things, so Amy's one of those people, as well as Matt, who get in there, they're all gowned up and have to work the entire day under these super uncomfortable conditions. But they're doing so to move the needle for us for the world. Mark Loeb is a guy who I've known since it seems like forever. He's one of the most amazing clinical researchers and fundamental scholars that you'll ever come across in the world, let alone McMaster University. He's been one of these guys who's been able to develop multidisciplinary teams to be able to study complex problems like vaccination, like who should be vaccinated, how do you study those things, how do you develop a protocol so that you're getting the best bang for your buck when you're either vaccinating people or treating them for whatever it is. So whether he's working on flu with Hutterite colonies in the West or dengue fever in Southeast Asia, he's just been a remarkable clinical researcher. And he also has to see patients on a regular basis. So he's one of these guys who can absolutely do it all, and we're just blessed to have him here as part of our team. And finally, last but not least, is Leyla Soleymani, who is an associate professor now, in it's engineering physics at McMaster University. So she's an engineer who's developing these new technologies for us to be able to detect infectious disease threats in a way that is well beyond anything that we've been able to do. And so she and her team have developed new diagnostic tools for COVID, for example, but also have great application to other infectious diseases as we go forward. So wouldn't it be great if we had a single device that could tell you right away when you get up in the morning, if that scratchy throat is just because the air conditioner was turned on too much last night and you got dehydrated, or it's because you've got a rhinovirus or coronavirus or whatever it is. So these are the kinds of technologies that we're bringing together. So this is our amazing panel today, and we're going to hear a lot from them, and I'm going to ask, I believe it's Matt, to come up first and tell us about vaccine strategies. So take it away.
Great, thanks so much, Gerry, and thanks all of you for being here today. I can say, although the last 2 years I think have been very challenging for all of us, one of the immense pleasures that has come to fruition over the past couple of years is the opportunity that we've had to extend our cross-disciplinary research networks, in particular with folks at MIRA, but also I've had the immense good fortune to work very closely with all of our other incredible panelists today as well. And so I know this will be a really interesting and thought-provoking session. What I'm going to tell you about today is some novel active and passive vaccine strategies specifically tailored to addressing challenges we have in protecting aging populations. And a lot of this work, as Gerry introduced, really started when I was a postdoctoral fellow, and we began thinking about how to make better influenza vaccines, and since that time has really evolved to extend to things like coronaviruses as well. So as many of you probably know, pandemics are unfortunately not new to us. Between 1918 and 2009, we experienced 5 flu pandemics, the worst of which was the 1918 Spanish flu. It resulted in the death of 3 to 5% of the global population. If we extrapolated the population in 1918 to the population today, that would amount to 370 million deaths in a 2-year period. So when people sit back and reflect on how bad COVID-19 has been and ask me, could it be worse? It could absolutely be worse. And this really underscores the need for us to think intrinsically differently, not about how we respond to infectious threats, but how we prevent them. The most recent flu pandemic, of course, was in 2009. Many of you will remember 2009 H1N1 swine flu. But when you remember it, you probably won't remember much encumbrance on your everyday life. I think sort of the most that pandemic impacted us was some signs in washrooms reminding us to practice good hand hygiene. And if you asked people in 2009 what the next pandemic would be, I suspect many would have guessed it would be another influenza. And what we've experienced also underscores, I think, a need for us to be very modest in understanding that we don't know a lot about what infectious disease challenges we might face in the future, and so we need to be prepared on all levels and in a very holistic way. The reason that response should not be our default way to approach controlling infections is illustrated by this graph, which in blue shows the number of ILI cases during 2009. In red, it shows the number of pandemic vaccines shipped, and in green shows the number of pandemic vaccines administered. In 2009, we'd been developing flu vaccines globally for over 40 years. We have massive global infrastructure to do this, in contrast to this current pandemic where we have never had a coronavirus vaccine before. And even with massive global infrastructure and complete know-how in terms of how to make new flu vaccines, we could not respond quickly enough to blunt that pandemic. And that's why we need vaccines capable of protecting us before pandemics emerge. If we look at the people who are most impacted by our failure to prevent infectious diseases, as we heard earlier this morning, it's disproportionately vulnerable individuals. In the context of flu, if we just look at age demographics, the people most at risk are at the extremes of their life. The very, very young, as you can see in the 0 to 4 age category, and in the older age category. And this is a problem because those demographics also present unique challenges to effective vaccination, because there are intrinsic differences in the immune systems of neonates and the elderly that make protecting them challenging, despite the fact that they're at the highest risk of severe outcomes. Over the last few years, this recognition has led to enhanced seasonal influenza vaccines for older adults. So there's now a high-dose vaccine, which is administered in long-term care, and there's also an adjuvanted vaccine. And if you look and compare the relative effectiveness of those vaccines against sort of the standard flu vaccine, you can see that they are considerably better in protecting older adults. But still, this leaves us unprepared for emerging pathogens. And even this relative effectiveness still results in overall fairly modest protection on an annual basis against flu. If we look at COVID-19, we've seen what I really think is a modern medical miracle in terms of how fast and how effective we were able to generate a new vaccine. But in contrast to what I think many in the general public were led to believe, the simple approval of these vaccines has not been a panacea that has ushered us rapidly out of the pandemic. In fact, if you look past the hashed line when the Pfizer-BioNTech vaccine was first approved, you can see there's still been major morbidity and mortality. If we look at COVID, one of the things that distinguishes it from flu is that there's some very important differences in who's most at risk. If we look at cases overall, the majority of cases are actually in younger people. The older adults have a relative paucity of overall cases. However, if we look at deaths, you can see that the relationship flips totally upside down. By far, older adults are at most risk, and in some ways, very fortunately, unlike flu, young children are not typically a high-risk population. One of the reasons that vaccines have not rapidly ushered us out of the pandemic is, of course, because we've seen the emergence of variants of concern that have mutated in ways that make current vaccines less efficacious. And so we need to get out in front of this virus if we want to effectively protect the population in the future, not only from variants, but from spillover by potentially novel coronaviruses. And so I hope what this underscores is that we need to turn our focus away from response and instead really harness our energy on prevention efforts. So I'm going to tell you just a couple of little vignettes today. The first story is about this massive collaborative effort at McMaster to generate a next-generation COVID-19 vaccine. So when we're exposed to a pathogen, the way that we're exposed profoundly impacts the quality of our immune response. When we get an injection in our arm, what's called a parenteral vaccine, we make lots of antibodies that circulate in our blood, T cells that circulate in our blood, but not much immunity in our lung. By contrast, if we get an infection or a vaccination in our lung, the quality of our immune response is much different. We get local resident cells capable of protecting us. We get different types of antibodies that are specific to mucosal tissues, and that type of immune response is specifically designed to protect us from mucosal pathogens. Our propensity to vaccinate people parenterally, I think, is really a carryover of the original Jennerian smallpox vaccines, where what was done was scabs from smallpox patients were used to so-called scarify naive individuals who had not been infected to serve as a vaccine. So basically, this smallpox scab material was sort of scratched into the arms of healthy people to protect them from getting disease. However, hundreds of years before the Jennerian vaccines, in China and probably in India, we were already vaccinating people mucosally through what's called insufflation. Essentially, it was the same idea. Smallpox scabs were crushed up, but then they were blown into the nose of healthy people in order to confer immunity directly in the lung. And so, in thinking about how to make better vaccines and how to make vaccines that can protect us more broadly, often what we do is we focus way too much on antigens, on pieces of pathogens that don't change, but we don't think so much about mode of administration. So when this pandemic first started, what we recognized very quickly was that coronavirus is a lot like flu in that it's capable of mutating very rapidly. All current vaccines targeted only the spike protein, and the spike really likes to mutate. And so if we were only going to target spike, we would be in the same place we are every year with flu, constantly chasing the evolution of the virus and never getting out in front of it. So what we decided to do was include two other antigens, nucleocapsid and RDRP, the viral polymerase. These are pieces of the virus that are only expressed inside of cells, but that T cells recognize really effectively and that don't change very much. These antigens are highly, highly conserved among all variants and even in other coronaviruses that only exist, for example, in bats, SARS-CoV-1, and MERS coronavirus, which is extremely pathogenic with mortality rates that can sometimes be as much as 50%. Contrast that to well under 1% for SARS-CoV-2. The first thing we wanted to do was test whether where we delivered this vaccine mattered in terms of the immune response. So we took mice and gave them this vaccine, which is delivered in an adenovirus vector, either in the nose to stimulate respiratory immunity or via injection in the muscle like we normally administer vaccines. And what you can see in the IN group, the group that got the vaccine in their nose, is that their responses, their antibody responses, were much, much stronger when the vaccine was given in the nose relative to in the muscle. And we also specifically generated antibody-producing cells in the lungs of those animals that weren't present when we vaccinated in the muscle. That's all fine and good, but of course what really matters is, does this differential mode of administration protect better when we're infected? And so in our level 3 lab, we experimentally challenged these vaccinated animals with SARS-CoV-2. And what you can see in dark blue is that the animals that received the vaccine in their nose were totally protected. They didn't experience any weight loss, and there was massive multi-log reductions in viral titers. However, the exact same dose of the vaccine given intramuscularly resulted in severe disease and lots of virus present. If we went and tested whether our vaccine was still effective against immune-evasive variants of concern, as you can see, the beta variant was one of the most immune-evasive variants before Delta and Omicron emerged, and our vaccine was completely protective against beta. We've since gone on to confirm this against Omicron, and again, we see the same thing. So this vaccine is fully protective against all variants of concerns that have emerged to date. In addition to thinking about sort of the unique cellular responses that are present in the lung, our group has also done a lot of work looking at how specific types of antibodies in the airway protect us. IgA is an antibody that is specifically enriched at mucosal surfaces like the lung and gastrointestinal tract. But we actually don't know very much about how IgA protects us from infection. There are two ways that antibodies can confer protection. The most straightforward way that we all usually think of is antibody binds to pathogen, and now pathogen can't enter our cells or adhere to our cells and gets eliminated. But the other side of antibodies, the Fc region, can also flag immune cells to come and eliminate infected cells after a pathogen has entered them. But we're only beginning to understand how that works, and we really had no idea how that worked for IgA. So Anastasia, an excellent PhD student in my lab, wanted to ask, how does IgA protect us from viruses? And what she found was that when viruses were coded in IgA, those complexes of antibody and virus could be recognized by neutrophils. And they stimulated neutrophils to undergo this cool form of programmed cell death called mitosis, whereby the neutrophil extrudes its DNA, virus sticks to that DNA, and then becomes inactivated. This didn't happen, though, with IgG, the antibody that we normally stimulate when we inject vaccines into the arm, and also the antibody type that's most prevalent in our blood. Importantly, this phenomenon was broadly true. It didn't matter whether we used SARS-CoV-2 or influenza or HIV or even autoantigens like citrullinated fibrinogen, which is an important autoantigen in rheumatoid arthritis patients. When IgA binds to these things and neutrophils see that, they all undergo mitosis. And what was really cool is Hannah was able to show that this mitosis led to both trapping and killing of the virus. So this mucosal vaccine is now in phase 1 clinical trials here at McMaster. We're enrolling participants who have been vaccinated with 2 or 3 doses of mRNA and are now in the process of preparing for the phase 2 portion of this study. The other thing I'm going to mention just very briefly is passive vaccines, which is another important part of our arsenal of how we protect aging individuals particularly. So I told you that antibodies can flag cells of the immune system to eliminate infected cells. Ali Zhang, an MD PhD student in the lab, discovered this really interesting phenomenon with monoclonal antibodies that are capable of recognizing essentially all strains of flu. He added monoclonal antibodies to flu-infected cells, and then on top of that, he added oseltamivir. Oseltamivir is an antiviral for flu, and it blocks this enzyme called neuraminidase. And when he did that, what he saw was that the addition of oseltamivir made immune cells better able to eliminate the infected cells. So we then asked, well, what would happen if we gave this antibody and drug as a combination therapy in flu-infected mice? And so we did this in 2 ways. Monoclonal antibodies can be used to prevent infection, but also treat it. So Ali either gave the antibody and drug to mice prior to becoming infected, or as a therapeutic after becoming infected. And we wanted to know, is the combination therapy better than either monotherapy alone? And what Ali found was both in the context of prevention and in the context of treatment, this synergistic relationship between an antiviral and a monoclonal antibody protected completely against lethal influenza. One of the issues, though, with monoclonal antibodies is that they're very difficult to deliver. And the people who need them most are honestly people living in communal care settings. Most of the time, the way we deliver these things are at infusion clinics, like I'm showing here. But infusion clinics also treat people with severe autoimmune diseases. And those people can't be co-housed with someone who has COVID. And so one of the real limitations in effective administration of monoclonals is how do you actually get those monoclonals to people who need them the most? One easy way to do that would be, again, to deliver them mucosally, because it's very safe and it could be done easily in long-term care, communal living settings, doesn't require the expertise of a hospital. So what we've done is we've given monoclonal antibodies to the respiratory tract of mice prior to infection, either 1 day, 3 days, 7 days, or up to 2 weeks. And what we see is that, amazingly, antibodies are capable of completely protecting these animals as far out as a week after administration. So, in summary, what I hope I've shown you today is that prevention really needs to be the major focus of how we protect populations from infectious disease, because we can never respond quickly enough. Older adults are an especially high risk group when it comes to respiratory viruses, but they also present real challenges when it comes to protecting them with vaccines. We need to avoid only focusing on the antigen when we think about protecting people, and also think about how do we administer drugs or vaccines in a way that's going to make them work best. And finally, we need both conventional active vaccines as well as passive vaccines to effectively protect these populations. So thank you very much, and I will turn the floor over to our next speaker. Also, I should say that this was a huge collaborative program, obviously, and there's a tremendous number of people to thank for all of the work I showed you today, most notably my lab, of course, who've been the real heroes in the CL3 lab through the pandemic. So thanks very much.
Absolutely. Okay, thank you, Matt. And Amy, tell us about humanized mice. Not a horror movie.
Okay, thank you for giving me the opportunity to talk here today, and I'm going to talk to you about humanized mice. And when you hear that, many people are like, isn't this an oxymoron? How can you have a humanized mouse? And really, in this case, it's when we give a mouse a human immune system. But how do we do this? So, of course, this is not the easiest process, right? So there's several key steps. And the first is that we need to have a source of human hematopoietic stem cells. For this, you can use things like umbilical cord blood, bone marrow, mobilized PBMCs, even fetal liver samples. And we use cord blood, and from this we isolate hematopoietic stem cells. The second key component is what kind of mouse are we going to use? So you have to use mouse strains that do not have their own immune system. Otherwise, they would reject the human immune system. So we have different immunocompromised mouse strains that lack B cells, T cells, NK cells, and we can add to these different transgenes to make them even better at being humanized mice. So we irradiate the mice, we inject the CD34 cells in them, we can do it intrahepatically in newborns or IV in adults. And then we wait, because as you might expect, it takes quite a while to reconstitute an entire immune system from hematopoietic stem cells. So it takes about 4 months almost before you get this full immune system. And you can see here we get really great T cell responses, B cell responses. We get some antigen presenting cells, particularly monocytes and some dendritic cells, as well as human cytokines in these mice. So why do we want to make humanized mice? It's a really important question. And as you might imagine by my introduction here today, one of their most useful applications is in infectious disease. So there are multiple human pathogens that actually require human immune cells to infect or to create a more human-like disease model. The most prototypical of those is HIV. It can only affect human immune cells, and thus normal mouse models aren't useful. But also Epstein-Barr virus, dengue virus, salmonella, HTLV, hepatitis C is the big one used, Zika and Ebola have all been tested in humanized mice and can replicate parts of the human disease. It's also used in allergy and autoimmunity, and in cancer. So I also do some projects in cancer with this in collaboration with Dr. Mossman. As I mentioned, you can have different models for this. One of the models that we like to use is one of these immunocompromised mice that actually has human HLAs in it. And then we can match them to the hematopoietic stem cells, which gives really great immune reconstitution. So our baseline strain is in blue, I think here. Okay, no, it's in black. Sorry, the colors look different up there. Is just our baseline immunocompromised mice, and our drag A2s have the human HLA. And this is showing how many human immune cells we can get in these mice. And you can see the drag A2, it does improve the levels of human CD45, which are immune cells, in the blood. But what kind of immune cells are there? Because that's very important when you're thinking about what types of immune studies you want to do in them, and in particular, if you're thinking about how to use them for vaccination studies. You can see we get high levels of overall human CD45 T cells, but we also have lots of T cells, B cells, and we do see higher levels of the monocyte macrophage lineage as well in our HLA transgenic mice. Now, as I mentioned, HIV is one of the main uses for humanized mice. So at this point, this is still global pandemic with 37.7 million people affected. And it's a pretty steady number of new infections each year at about 1.5 million, and just under 1 million deaths per year still. And you'll also note that the demographics have changed quite a bit over time, and women are actually a higher proportion of individuals living with HIV at this time point. So one of our first steps was to use these models to see if we can appropriately model HIV. So here we take our humanized mice and we can infect them. We infect them either intravaginally to mimic infection in females, and we can also do systemic infections to mimic drug use exposure. So within these mice, we can get high levels of HIV in the blood. It's, on the left, it's HIV copies per mil. We do qRT-PCR to see this. So we get high expression in the blood over time. In the vaginal wash from these mice, we can see that it peaks at the beginning, and then as the virus disseminates out to the body, it decreases. And we get the prototypical loss of CD4 T cells, right? So we start with high levels of CD4 T cells, and then as the HIV progresses, we see a massive decrease in CD4 T cells. So these are very good models of human disease. Now, to shift gears, tuberculosis, we've heard about a little bit here, right? Mycobacteria, it's caused by Mycobacterium tuberculosis. This is spread via aerosol and is very infectious. You need very few CFUs to become infected. Luckily, the majority of people that are infected develop a latent infection. We are able to control the TB bacteria by forming granulomas in the lung. But everyone still has a 5 to 10% risk of developing active TB over their lifetime. 1/4 to 1/3 of the world's population actually has Mycobacterium tuberculosis. So this is a huge issue. And in 2019, 10 million people developed active disease. 1.4 million people died from TB. And it's actually one of the top 10 causes of death worldwide. And even more worrying is the fact that multi-drug resistant and extensively drug resistant isolates of TB are increasing over time and may become a big factor in the future. So can we model TB disease in humanized mice? And so here we again take our strains, we infect, this is done in the level 3 facility, we use a virulent strain of TB to infect intranasally to mimic the aerosol exposure that humans receive, and we wait for endpoint, and then we can look to see how disease develops in these mice. Here we see log CFU, or colony-forming units of Mycobacterium tuberculosis in the lung and the spleen. So in the first column, it's in the lung. You can see in both mouse models, we get very high levels in the lung, which is common for TB. But we also get dissemination from the spleen, which is also a key component of TB. Here you can see just an example of an uninfected lung and then an MTB-infected lung. And what you see within that lung in the histology are granulomas forming, trying to contain the MTB, just like we see in human disease. And the granulomas that form are actually very human-like and show characteristics such as foamy macrophages. We get I'm going to show in the next picture. I'm jumping ahead. Here I zoom in on a granuloma, H&E on the left. You can see the necrotic core, which is characteristic of human granulomas. AFB stands for the actual mycobacterium. So there you can see that they're centralized in the core of the granuloma. And the CD4 immunohistochemistry, I don't know in this light if you can see it nicely, but the brown cells surround the granuloma, and those are actually immune cells trying to contain it. So all this is occurring in our humanized mice, indicating it's a good mimic of human disease and can be used for future studies. So Gerry did mention I do concentrate on HIV-TB co-infection, and we've just established this, but I'm not going to concentrate on this here because I wanted to highlight some of the work we've been doing with vaccines in these mice. So vaccination in the elderly. So the general consensus is that the elderly do not respond as well immunologically to vaccination as the young, but it's not super well studied in the literature. The best evidence comes from the flu vaccines and is more recent. as they're often excluded from clinical trials. And COVID has really changed that. I think we've realized the importance of looking at it in the elderly, responses in the elderly. But you might ask why, and I think, again, Matt kind of touched on this a bit, but the immune system changes over time, right? So now it's thought that we have something called inflammaging, and there's chronic inflammation that slowly over time leads to changes. One of the main populations that are involved in this are T cells. We get CD8 T cell senescence, reduced repertoire, so reduced ability to respond to new threats or new vaccines per se, increased T regulatory cells, which aren't as useful in vaccination. In terms of B cells, Matt again talked about the isotype switching, how we need different isotypes for the best responses induced by vaccines, and this is impaired in the elderly. And dendritic cells and macrophages, we talk about these as antigen-presenting cells. And these are really key to vaccine responses. And they've often shown decreases in activation as well as ability to phagocytose and process antigens. So you can see with all of these that we might need to think about how we can improve immune responses to vaccination in the elderly. And I got a great introduction to this. One of the main, there's multiple ways we can do this. One is altering your strategy of vaccination, right? Now we all know about mRNA vaccines versus protein subunit vaccines versus vector-borne vaccines. And these can all cause a differential immune response. Secondly is altering the route of vaccination. And definitely at McMaster, we're a very big proponent of the mucosal vaccination, of getting the immune response where we need it most. And this would most likely work, again, in the elderly. And the last is using different adjuvants or immunomodulators to increase responses. So we've collaborate with the Zhou Lab, who have created TB vaccines kind of similar to what you saw Matt present for the COVID vaccine. So it's actually a replication defective adenoviral vaccine. It's ChIMPAD. It contains 3 TB antigens, just like the COVID vaccine they've designed has. And this targets different stages of the life cycle, so it should be more effective than just targeting one. And of course, we do administer it mucosally, and it's been shown we believe it will target both innate and adaptive immune responses, which would be beneficial. So to test this in my humanized mice, we've already shown they're good models of TB, right? So once we've shown that they can do or replicate disease, we can use these mice to do challenge experiments and determine how well vaccines can protect. So in this case, we give the vaccine, we wait 4 weeks for the adaptive immune responses to occur, and then we can actually challenge with TB. We can also look at immune responses before challenge to see what's happening. And that's in my first slide here. We're looking at just what happens in the lungs when we have given this vaccine and we wait 4 weeks. So you can see the unvaccinated mice are in black here, and the vaccinated, again, those 2 groups I have of humanized mice are in pink and green. And you can see when we take out T cells from these mice, show them TB antigens, and say, can you respond by making the cytokines that we need for protection? You can see that we can get interferon gamma production and TNF-alpha production. On the left, we have percentage of CD4 T cells that are able to do this, and on the right are actual numbers of cells in the lung. So we can see we have both of those increased. And in addition, we have CD8 T cells, although a little less so. But it is known this vaccine targets CD4 T cells particularly. But the big news comes, and I'd like to thank Sam, who's here today, for rushing this data, because we just got it last week. The big bang for your buck comes when we give these mice TB and see what happens if they've been vaccinated. So in our top row, we have our unvaccinated mice. Our bottom row, we have our vaccinated mice. And the gross is not because it's gross, but because it might be a little bit. Sometimes when we pull these lungs out, we're like, whoa. Okay, so you can see in the unvaccinated, there are a lot of granulomas. The white nodules you see are granulomas forming. And our vaccinated, we have a nice pink-looking lung. When we section these and do H&E, again, you can see almost the whole lung in the unvaccinated is granuloma. and there's just small granulomas found in our vaccinated mice. And AFB, again, shows the mycobacterium themselves, and you can just see the mass of mycobacterium in the unvaccinated lung versus the very isolated pockets in our vaccinated. And when we look at the number of bacteria actually in the lung, on the top there and the spleen on the bottom, you can see we get over a 2 log reduction, which is quite a bit in terms of TB. TB is a very hard pathogen to prevent. So 2 logs difference is actually showing a very high efficacy of the vaccine. And again, we get decreased dissemination as well to other sites like the spleen. So overall, I hope I've shown you that humanized mice can really recapitulate many main features of human infectious diseases. I've shown you here HIV and TB, but there's others. Immunization and challenge studies using humanized mice models can be used to examine vaccine efficacy in human immune cells. So that's the difference than using these in mouse cells. It's actually showing that the human immune cells have the capability to respond to your vaccine. So really, I think these mice act as great preclinical bridges to facilitate translation of basic research that we have into the clinic, right? This can save us time and money by really seeing how, or developing the best vaccine that we can before going to clinical trials, say. And I would like to thank all the people who participated in the work in my lab, Maddie and Jack in particular, in the work I've highlighted here, my collaboration with Zing and Sam Afghani from the lab have been instrumental to a lot of the work and help in the level 3 facility. And I think that's, and my funding institutes, IIDR in particular for this work. Okay, thank you.
Thank you, Amy. That was great. A lot of questions for you, but I'll hold them until later. And, but Mark, thank you.
Okay, great. Thank you so much. Thanks, Gerry. Thank you very much for the kind invitation to present today. So I'm not going to focus so much on my own research. I was asked to give you a talk about vaccine-preventable infections just generally in older adults and from a clinical perspective. So that's what I'll do. And I'll first just start by describing some of the most common vaccine-preventable infections in seniors. And I'll review some of the key gaps in the evidence, and in so doing, describe some of the challenges methodologically in terms of how vaccine efficacy is evaluated, and talk a little bit about the specific challenges in older adults. So I'll first start with influenza. As you know, influenza is a vaccine that has to be given annually. And some of the sort of seminal data on the epidemiology of influenza came from Arnold Monto, who did a bunch of studies in the late 1980s and 1990s, where he looked at a population in Tecumseh, Michigan, and said, we're going to swab this whole population and see who gets the flu and who gets complications. So Dr. Monto found out, as you could see on the left-hand bar diagram, that the attack rate in children is the highest. And frankly, that's why we've been doing studies in the Hutterite community, doing randomized controlled trials to vaccinate the children to look at indirect benefit. But on the right-hand side, in the middle bar diagram, you see that the hospitalization rates, as Dr. Miller had suggested, are highest at the extremes of age. So it's always a key theme in infectious diseases, that it's either the young children and the older adults who end up with complications. And hospitalizations for flu is relatively common among children less than 5. However, the deaths are not that common in younger children, and the bottom diagram shows that the death rate is highest in the elderly, and that increases with age. This is a slide looking at influenza-associated hospitalizations in the United States, and the key thing is the the second column looking at rates of pneumonia and influenza hospitalizations, that you see as people age from their 60s, 70s, and 80s, there's a huge increase in hospitalizations, and that it's not only limited to hospitalizations for the complications of flu, but it also includes respiratory and circulatory hospitalization. So that's sort of another theme, the circulatory thing. So we've completed a randomized controlled trial that's, again, we've presented it publicly, showing that during flu season, if you get a flu shot, if you have cardiovascular risk factors, it'll reduce your risk of death. So that's in review. It was presented, I presented it at the American College of Cardiology meeting a few months ago in Washington. So in terms of influenza vaccines, I want to focus on the fact that in terms of prevention in Canada, everybody over the age of 6 months, it's recommended should get a vaccine. Some of the vaccines are trivalent, quadrivalent, has an extra strain of B antigen, and that's most important for children, probably less so in terms of adults, particularly in terms of the paucity of B lineages that are circulating recently. Live attenuated vaccine is for children, but the key ones, as Dr. Miller had pointed out, are the adjuvanted vaccine for older adults and also the high-dose one. And there have been randomized controlled trials comparing the adjuvanted vaccine, the high-dose vaccine to standard dose vaccine, and the evidence was about a 30% risk reduction with the high-dose vaccine. But if you look at the randomized controlled trial generally for influenza vaccine efficacy in the elderly, it is a dog's breakfast. There are studies, this is just an outline from all sorts of populations, psychiatric hospitals, where the vaccines are, vaccines no longer on the market. The outcomes they use, the designs were flawed. Many are not blinded. They failed to conceal allocations, basically real no-nos in the randomized controlled trial world. The outcomes also, often they're not PCR confirmed, so we really want to see, at least at some point, some virological outcomes. And many of these were serologically based or just clinical based, and that's sort of a limitation. The vaccine efficacy is relatively variable as a consequence. And in the 1990s, I'd say, there was a series of studies showing tremendous bias in estimates of influenza effectiveness in seniors. There was all sorts of studies showing that you give a vaccine, a flu vaccine, to a senior, and their risk of death would go down by 40%. Or the risk of hospitalization. So this was actually an interesting study by Lisa Jackson from Seattle, and what they did is they compared individuals who died from either flu or pneumonia or were hospitalized, and they compared those to seniors that did not have those outcomes, and they looked at the proportions of exposed to influenza. And they sort of did something sneaky because they said, okay, well, let's do this comparison, but we'll do it before the virus is actually circulating. And they found a tremendous reduction in all-cause mortality and in pneumonia and influenza hospitalization. And the only explanation is a healthy person bias, that is, seniors who were healthy to begin with were selectively getting vaccinated. Obviously, there was no biological plausibility. And they did a second study looking at functional status, and when they adjusted for the functional status of the participants, this effect of reduced mortality and pneumonia and influenza hospitalization just went away totally. So there have been a lot of biased studies. One sort of, I guess we could call it an innovation methodologically, has been the test negative design. So when you're talking about trials or studies to evaluate vaccines, you're really left with either randomized controlled trials, which are the gold standard, or observational studies. And for observational studies, I'd say the gold standard is the test negative design. So this is a type of design where an individual who has let's say if it's for flu, they have a flu-like illness, they're tested, and if they have flu, they become a case. If they don't, they become a control, and you look at the vaccine exposure in both groups. Now, there are some limitations to this design, but it's clearly better than those cohort studies that were being done in the 1990s, where you have at least probably 10 to 15 studies showing the selection bias. Then there's, vaccine against confirmed influenza caused by strains similar to vaccine components. So this was a sort of a vaccine trial of an adjuvanted vaccine comparing. This was the high dose rather. This was the high dose versus standard dose vaccine. So this is the trial from the New England Journal that showed about a 30% risk reduction if you have the high dose vaccine. One of the limitations, though, is that although the high dose is a randomized controlled trial, the adjuvanted vaccine, there's never been an RCT. It's really based on observational data, and there's never been a head-to-head comparison. So probably for the last 6 years now, we put together a proposal to do a cluster randomized controlled trial of every nursing home in Ontario, where we'd randomize them, the residents, to either a high dose or an adjuvanted vaccine. And it's actually powered as a non-inferiority study to show, well, probably there's no difference. And it's certainly important because the high-dose vaccine is about 5 times more expensive than the adjuvanted vaccine. So we have the funding, we could do it internally, we have everything ready to go, but the okay from the ministry. And every year, and we go to them, and it's sort of like Charlie Brown and the football. We say, can we do it this year? And they pull the football. And right now, we've made the proposal to them again, and we're just waiting to hear back. But I think it would be a very important study to do, and we would embed ILI, we'd embed RT-PCR confirmed influenza within the design. So we're waiting about that. So just moving on from influenza, Streptococcus pneumonia or pneumococcus is another really important pathogen in older adults. There are 90 different serotypes, so the protection is by a capsule, and it's serotypes sort of specific. So when you have a vaccine, you're basically generating antibodies against specific serotypes. And the age distribution in terms of complications is very similar to the flu. You have it at the extremes of age. The very young and the very old are at risk for complications. And when you look at independent risk factors for pneumococcal disease, you can see that, of course, people with comorbidities are at high risk, and unfortunately only about 30% of those actually have have uptake with the pneumococcal vaccine. It's relatively low, but residence in a nursing home is an important risk factor. Again, CDC has done studies comparing seniors in long-term care facilities to age-matched controls in the community, and the risk of invasive pneumococcal disease about 4-fold higher in the nursing home. That means invasive disease means having it in your bloodstream, basically. So residents in a nursing home and really what you've heard about the changes of immune senescence all increase the risk for pneumococcal disease. So the elderly are at very, very high risk. There are vaccine studies. There's the PCV13, which is a conjugate vaccine that produces a bit of a better immune response than the 23-valent polysaccharide vaccine. And this large study that was conducted in the Netherlands, I'd say if there's any milestones in pneumonia in the past 10 years, it's been this, because they randomized thousands of seniors to either the PCV13 vaccine or control, and they found a substantial benefit in terms of the risk reduction in pneumonia, about almost a 50% risk reduction in community-acquired pneumonia due to vaccine serotype-specific strains. So that's really an important milestone. Part of the problem, or some of the challenges with pneumococcal vaccine, is finding out what to do next, because the serotypes are always evolving, and more vaccines are coming out. So there's a PCV13, there's a PCV20 that's coming out in the market. And it's always the challenge to say, well, what about the indirect benefit because of vaccinating children? So there's a very strong herd effect when you vaccinate children, and shown here that when children are vaccinated, even the invasive disease in older adults tends to plummet. So it's always a sort of it's almost a modeling type of question in terms of trying to predict the future and saying how can vaccines be most appropriately used, both in children and adults, to try to reduce the burden of disease in older adults. So moving on now from pneumococcus is zoster. So the varicella zoster virus causes two forms of disease, either chickenpox, most of you are familiar with that, or herpes zoster or shingles. So the primary infection results in the characteristic rash of chickenpox. And the problem is that the virus establishes a permanent latency in the dorsal root and cranial nerve ganglia, basically. So it basically hangs out in your nerves, and because of immune senescence, or if you happen to be immunocompromised or get on steroids, it'll reactivate and it'll just crawl up back to the skin. The virus will go up the skin and cause this rash that you can see, and this can happen years later. And the lifetime risk actually is substantial. It's estimated to be about 30%. The burden is high. 1 million cases in the US, divide that by 10, 130,000 in Canada. Many hospitalizations, many visits to MD visits, and about over 15,000 cases of post-herpetic neuralgia. And that's one of the most dreaded complications because it's an excruciating, painful condition that's defined by pain that occurs, that persists for at least 30 days after the rash. So it's estimated to occur in up to 27 of people with herpes zoster overall. So it's a real burden, and this is the most famous study from the 1970s, Hope Simpson, looking at the incidence of both herpes zoster and post-herpetic neuralgia. And you can see that it starts after the age of 50, but clearly after the age of 60, the rates of both zoster and post-herpetic neuralgia go up quite quickly. So in terms of vaccine, the initial vaccine was tested in the VA population, very large randomized controlled trial, about a 50% risk reduction, which is great, reduced post-herpetic neuralgia almost by 70%, and decreased the burden of illness by 61%. So a really important milestone. And that's been superseded, and I'll just skip this one, and that's been superseded by the recombinant vaccine, which is known as the Gillgrass vaccine right now, which is very incredible efficacy, over 95% efficacy in older adults in terms of reduction in zoster, also substantial reduction in post-herpetic neuralgia. The challenge with the recombinant vaccine, the Gillgrass vaccine, is the reactogenicity. So there's a higher incidence of grade 3 symptoms in this population. Really, this is defined by symptoms that might interfere with functional status. Clearly, for me, the sort of risk to benefit is for this vaccine is clearly in the benefit realm, but the uptake is relatively low. It's around, I think it's under even 30% in the general population, and it's surprising because it is such a good vaccine. Moving on then to the last one is SARS-CoV-2, and I'll just say just a few words about that. You've heard Dr. Miller talked about that, but the burden of COVID-19, particularly in the long-term care facility, I'm not going to go on about that, but huge burden. And the vaccines really change things dramatically in this population in terms of reducing, particularly reducing deaths in the population. There's still a lot of questions, though, I think, that have to be addressed related to, booster doses, 4th, 5th doses that we don't know. And the most important design trial are randomized controlled trials, and to the best of my knowledge, there is no randomized controlled trial ever done in a nursing home. So we actually started trying to do a trial in Ontario, looking at the effect of a 3rd dose, and when that was being introduced, and then we got Health Canada approval, and about the day that we got approval from Health Canada, we found the government was switching to that. So we went to Alberta and couldn't do that. So we've gone all over the world to look for jurisdictions to do an RCT, and right now we're doing an RCT actually in Poland because they've been very slow to roll out the vaccine in this population. So they have a population of individuals that have had their 3rd dose, so we're randomizing them to the 4th dose using PVC-13 as a comparator. So I think there's a lot of things that could be doing. Again, looking at neutralizing antibodies, and Dr. Miller is involved. So I think that it's going to be an interesting study. So I'll just summarize then by saying that seniors are at very high risk for infections and the related complications. Influenza, pneumococcal disease, zoster, COVID, of course, are very preventable vaccine-preventable infections. And I think my message would be that we need to do more clinical trials to answer very key questions. Thank you.
Thank you very much, Mark. And last but not least is Dr. Soleymani, who will tell us all about diagnostics. So how do we anticipate using technology?
Well, thank you for having me. I'm really excited to be here to talk a little bit about the engineering technologies we've developed for rapid diagnostics. So I wanted to start with acknowledgements just so that I don't rush at the end. So this has been a real team effort with Ying-Fu Lee, who is a nucleic acid researcher, and Todd Hoare, who is a chemical engineer working on biointerfaces, and a clinical infectious disease team with Debbie Amamura, Marek Schmea, and Bruno Selina. Of course, for the COVID work, this team doubled, and we got support from people like Matt Miller, Karen Mossman, Cynthia Ballion, and John Brennan, and the real lab heroes are also listed below who've actually conducted the work. So really, what is the problem we've been really trying to solve for the past couple of decades? Is that even today, when you go with symptoms to see a healthcare provider, a lot of times you're asked about the symptoms, about history, but then if you have infectious disease symptoms, you are either given a prescription right there and right now, or you're asked to go back home, come back if the symptoms change, or when the lab results are delivered. So we thought, we're advised by clinicians that it's really important to have tests that right at that consultation can tell us, what's causing these symptoms. Is it an infectious pathogen? If it is it a virus? Is it a bacteria? If it is a bacteria, what species and how well it's going to respond to antibiotics? So there are tools available. The question is, why is it hard to do rapid diagnostics even today? I mean, some of it has to do with the inherent slowness of bacterial cultures when you're thinking about bacterial infectious disease diagnostics, but there are techniques that are not inherently slow. It's just that they have to be operated at a centralized facility, like PCRs of the world, ELISA's mass spec, and they have to, you need that lab infrastructure, you need that lab technician. So there are solutions already available. For example, there's bacterial dipsticks, there's urine dipsticks, there's lateral flow assays, but they're not perfect. So in some cases, like the dipsticks, they actually don't do bacterial they don't do bacterial identification. They will not tell you if the infection is caused by club pneumonia or E. coli, for example. And then the lateral flow assays that we've gotten really used to working with them, they're also not perfect. There's sensitivity and specificity challenges, but also a lot of these technologies are not multiplex and they're not universal. So you have a set of technologies or a group of technologies that are for bacterial infections, another group that are for virus infections. So I know Matt talked about universality. It's also important to have universal tools here as well that can be applied to all kinds of targets. So these are the things we're working on. And our approach to solving this problem is using biosensors or biological sensors. And so really a lot of the tools that are available right now are the benchtop, laboratory scaled instruments that you have on top, but we want to move to more and more devices like the one I have on the bottom, which is the continuous glucose monitor, which is a wearable, or it's in vitro diagnostic analog, that's the handheld glucose monitor. And by no means the new tools are going to replace those high throughput laboratory instruments, but there are settings where the healthcare system can truly benefit for having access also to a wide range of tools for in vitro and continuous diagnostics. So you might ask, what are biological sensors? So these are devices that bring together biorecognition with signal transduction to analyze biologically relevant analytes. So what that really means is that you need something that's going to, that I have on top basically up here, that is going to really grab into the target analytes that you want to detect and identify it very specifically. And then you want an engineered device that's going to really tell the user, yeah, I grabbed that, and you want it to say that with as much accuracy as possible. And so we are really working, in our team in all of those levels. So from biorecognition, the biointerface, which is between that biorecognition and that engineered device, and also on the engineered device. So I'm saying here that infectious disease sensing using functional nucleic acids and electrochemical readout, and you might say, why? I don't understand any of that. But the reality is you have been really interacting with both of those things, and I will talk about that. So, we're all familiar with single-stranded DNA or DNA in general. That's the building block for genetic materials. And I started my career really building diagnostics with single-stranded DNA to really fish out its complementary target, looking at bacterial DNA or messenger RNA, and et cetera. And then later got involved with functional nucleic acids like DNA aptamer or DNAzyme that we have on this slide. These are different structural motifs of DNA that can build themselves into specific shapes, and then they can target specific target molecules that don't have to be nucleic acids. It could be proteins, it could be small molecules. And so, for example, DNA aptamer can conform to a secondary structure that can tag its own target, kind of like Legos fitting into each other. And then DNAzymes are artificial analogs of enzymes that can catalyze a reaction, but they're not protein enzymes, they're DNAzymes. And we have a really nice picture there with the hands, so just kind of drawing aptamers as hands that grab into these spike proteins on the surface of the SARS-CoV-2 that really shows how they work. So the functional nucleic acids can self-assemble themselves into shapes that can grab onto biomolecules of interest. And just want to, because I guess the title of my talk was Detecting the Known and the Unknown. There's these functional nucleic acids are actually selected and isolated through a process called in vitro selection, where I know Ying-Fu Li often talks about it like American Idol. So you have a lot of these nucleic acid contestants that come in, but only a handful of them. So you start with 10 to the 15, and only a handful of them are the right fit. And you want to identify, amplify, and isolate those. And that's what's done through in vitro selection, and it can be done using known biomarkers, known proteins, or it can also be done, there are methods to do that using biomarkers that we don't know what they are, but we have access to samples that have, that are from infectious patients, for example. So this is really important, again, if we want to be prepared, like Matt said, we want to do, we don't want to have long response time. So if there is an emerging threat, you can very quickly go through this process and identify those American Idols. Okay, so and then I said you're more familiar with both nucleic acids and electrochemistry than you think. So if you think about the glucose monitor, that's an example of an electrochemical biosensor. You might have not known that, but you've seen people use it. And the way it works, you have your handheld reader. That's your electrochemical reader. And you have these test strips. And on the test strips, you actually have glucose oxidase that catalyzes the oxidation of glucose, and then it generates hydrogen peroxide that you can detect electrochemically through this reader. So this technology has been around, commercialized for multiple decades now. And one of the reasons, I mean, of course, there's a great market for a technology like this where diabetics have to measure their blood glucose level multiple times a day, but also glucose is abundant, it's freely available, so the levels are high. And so a technology like this, you know, has been around for a long time. So what we want to do, and what we have been doing really is to develop similar technologies that actually interface with the cell phone. And you can see a picture of one of our devices. You have a cell phone, there's a dongle, and then you have a chip that goes and gets plugged in. But now, instead of a glucose oxidase that can only detect glucose, you want to change that chemistry on the chip to include functional nucleic acids that can now, you know, again, take you to that universality. You can detect proteins, you can do small molecules, and you can even do DNA if you just use regular nucleic acids. And the way it works, you measure current, voltage, impedance using this really tiny USB stick size reader. You can do all of that and do diagnostics. Okay, I just want to show you some of the examples of these technologies. For both bacterial sensing and viral sensing, and of course, you know, in the near future, we're looking into doing both on the same chip. As you know, urinary tract infections are you know, one of the most common infections, and especially affecting the aging population. And what's typically done is urine culture, which has a turnaround time, even with rapid cultures, of overnight or in the order of a day. So we've developed a platform that you can see up here that can do, you know, sensitive and specific detection in an hour or less. So you don't need to do that culture. It's culture-free. And as such, using the combination of functional nucleic acids and electrochemistry, you can do this quickly. And what's really amazing is that, I mean, I was always skeptical whether we can do this when we look at such heterogeneous set of samples from urine, you know, especially with symptomatic patients. You have, you know, some that look like there's nothing at all, some that look bloody, some that have cast. But it looks like we're able to do the detection in an hour or less. There are some improvements we need to make with respect to specificity, because our test actually detects very low levels of bacteria in urine. And then, you know, why would this be useful? Again, I want to kind of loop back to the slide I started on, that if you look at the anti-biogram in the bottom, it basically tells you in any, you know, local environment, whether it's a long-term care, walk-in, it's a hospital in a specific region. For example, this is for Southwest Ontario. You know, what are the resistance rates to different bacteria? And it helps to know what species of the organism is causing the infection to prescribe the right antibiotic to the right population. And so our tool allows us to do that, to do the detection and identification in less than an hour, which, you know, it's a lot longer for the cultures, and it's not available with the dipsticks. So the speciation and identification, it becomes very important here. And I just wanted to also show you a snapshot of viral sensing. Again, we're combining now a different biorecognition element, an aptamer specific to this virus that I'm going to talk about, and we're integrating it again with that electrochemical reader. This time we decided to target animal diseases because, I mean, there is the economic burden, there is the food security issue, but also there is the transfer between humans and animals that we're hearing a lot about. And so again, we wanted to develop a saliva or a nasal swab test that can be used in animals, again, for rapid diagnostics. And a similar strategy works here. We're targeting the nucleocapsid protein of the porcine epidemic diarrhea virus. Yingfu Li's team has generated an aptamer through in vitro selection process, and we're able to again do analysis in an hour or less directly in pig saliva. And then you might wonder, how does this help us, or how has it helped us during the COVID-19 pandemic? I mean, fortunately, we've been working on detecting coronaviruses in pigs, and we've been developing these platform technologies for a long time. So then, when the pandemic happened, we basically had these chips, we had the reader, and we had the strategies to do signal transduction, and then Yingfu's team developed a range of aptamers that were selected both for the spike protein and nucleocapsid protein of SARS-CoV-2, and we developed a diagnostic test that can detect these targets directly in saliva. That's one of the important parts of our assay. We wanted it to be saliva-based so that the sample collection is easier to do. And so we developed a 10-minute test for COVID-19 in saliva that has a good sensitivity and specificity and in line with the requirements of a rapid test. And this platform is being commercialized by the company Zentech that is supporting a lot of this technology and our platform for current and emerging infectious disease diagnostic systems. And so the conclusion is that we have a modular technology that combines electrochemistry and these functional nucleic acids, and it's quite versatile. We can rapidly respond to new threats by really changing that nucleic acid component and somewhat our diagnostic readout depending on the levels of the pathogen and the matrix we're working on. And we're always looking to tackle the next big and important problem in this space. Thank you.
So that was great. So we now have the opportunity to ask questions, and this is one of the rare forums where I will also encourage comments and questions to our panelists. And I'm just wondering if anything comes to mind right away, bearing in mind. Claudia, you have your hand up, please.
Thanks very much. So thank you to all the speakers. Really, really interesting and fantastic work. And it's let me just say that, I read about all your stuff like on the Mac News and stuff, but you don't really have an appreciation for it until you actually see this. So it was really great to see all of this. I actually have a question for each of the speakers. I don't want to take the time, so like how many questions can I take? Just one?
Keep going
really?
until
right.
I tell you to stop.
So I'll go in order. Let me start with Matt. So really, really fantastic stuff. So you mentioned that two strategies for vaccination. I was wondering what your thoughts are on vaccination strategy to prevent zoonotic jump. So there are colleagues working in the US, working on transmissible vaccines, for example, for vaccinating mice so that prevent Lassa. I'm wondering what you think about that in terms of
so the issue of preventing zoonosis is really important, but also very tricky. I think the best example we have right now are rabies vaccines, right, which are bait-based vaccines and for which actually McMaster had a huge contribution because they are adenoviral vectored. Those are often deployed in relatively urban settings, and a challenge is reaching the reservoir. So rodents, which are an important vector for a lot of really high consequence pathogens, these transmissible strategies I think are really, really promising. There are, in certain cases, considerable regulatory hurdles to overcome. In terms of human health, though, my real feeling is one of the tricky things about designing vaccines that can protect against broad ranges of pathogens is that you can't test the efficacy of a vaccine against something which has not yet emerged. And so, over the last couple of years, I've been thinking a lot about, well, what's the regulatory framework for this? And I actually think the best thing to do are non-inferiority studies. I think we need vaccines that are at least as good as the current vaccines in protecting against what we know is out there. And then we need to use immunobridging studies. In some cases, there are human challenge models available, which are amenable to this kind of thing, to demonstrate their likely utility in protecting against broader ranges of pathogens.
Thank you. Since I'll just go to Mark because you just mentioned the part about trials. So, Mark, I'm curious why the Ministry of Health is holding up. What's the study?
I don't know. It's been 6 years. Every year we go for it. So I don't know. I don't find the reason. It's I'm not sure. It could be political because they understand it and they're very supportive of the question. And right now the federal government is providing high dose for long-term care residents across the province, but that donation is ending, right? So it'd be very important to do this. It's, as Matt mentioned, this is a non-inferiority trial, so it'd be very important. And in Ontario, it would be so, I'd say easy. It's never easy, but relatively not so challenging to be able to do this. Yeah.
Thanks. So my question for Amy, it's really interesting work. I met Honkwi Deng several years ago, who was doing similar work with the Humanized, the liver. I'm wondering, and he had a lot of issues around this notion of the chimera, that you're creating a chimera. I'm wondering if like the ethics components of this around creating chimeras, if it's been pretty straightforward, if you've encountered any challenges.
So, I would say there are some issues when thinking about how the mouse and the human cells are interacting, right? That's definitely on my mind a lot in my models. Sometimes we do get GVHD, or graft versus host disease, where if we have contaminating T cells, they can attack the mouse host cells. But some of my models do try to deal with those issues with getting mice that have human HLAs so that we can match them better and kind of overcome some of those limitations.
Thank you. And my last question, and then I'll pass the microphone on, is to Leyla. Really, really cool stuff. I'm wondering if your device, so you're thinking about like broader application, obviously lots of potential in low and middle income countries where point of care diagnostics are really, really important. Is this something that would be affordable? Is this something that, for example, like who can administer this? Could this be like a local community worker or healthcare worker? So I'm just thinking about what the possibilities are for applications in global health and LMICs.
Yeah, so I think one key area of application is resource poor areas, because the readers are reusable and they're $100 or less, and the chips are disposable in the single digit dollar values. So definitely, and the users can be trained to self administer the test. So it's a tool for both worlds, right? The low resource areas, but also we have, the COVID-19 pandemic tells us that we need it as much as anywhere else, right, as well. So I think it has applications, both in both areas.
Yes, question in the back.
That was fascinating. Thanks very much to all of you. I'm going to make a suggestion that one of the biggest pieces in why aging has been amplified at McMaster is because of the Optimal Aging portal. It's a portal that disseminates information on various levels that people at various levels can understand. Even to get an endorsement from my parents, neither of whom went to university, I think is pretty strong. So are there plans for an optimal immunity or infectious diseases portal on the table? Because surely all of you, you've done great jobs, really proud, it's great work, but the translation of some of these things and the knowledge myths and maybe even disinformation that you have to fight against it must be frustrating, or not, let alone to have the ball pulled out from under you by the Ministry of Health every year. But something on that level might begin to push people and change opinions or
So I guess I'll briefly tackle that in the sense that it's perfect timing, because one of our pillars in the application that Parminder and I are co-leading is exactly that, is how do we get this kind of information out to, and we're going to take full advantage of the existing portal and expand it as much as possible. But it's great to hear that your parents are using it. And really outstanding endorsement of that approach, because it's so vitally important, as you point out. I don't know if anyone else has that.
Yeah, I was just going to say, I think these resources are tremendously important, and yes, we've all been very frustrated by disinformation. I think one of the biggest challenges is it's one thing. It's critically important to present information in a way that's accurate and accessible. I think one thing we've learned during this pandemic especially, though, is you have to find really effective ways to draw people to those reliable sources. And I think that's the biggest challenge, because there's this echo chamber effect in social media, right? And so sometimes the people who need this the most will never find it because it will never enter their sphere. And so thinking about really creative ways of that level of penetration, I think, is the other key piece, in addition to having really accessible formats.
I can tell Parminder wants to.
I always leave them. I think adding to that, one of the things I was talking earlier on is the example where with the MIRA, we have partnered with Dixon Hall, hard-to-reach community. They don't have access to internet. A lot of people can't get that information. So working with community organizations, that become the hub, and translating that information to the people who can't get is one creative way to achieve that. And I think we do need some combinations of approaches to do so. Another thing is that the portal that does very well is one thing that it's really hard to keep up with misinformation. You can have all most credible information, but you can't. So the group that sort of spearheaded the portal, anytime there was a news story that was about science and was sort of iffy, we called it hitting the headline. So at the same time, we will release, okay, this is what the evidence in the literature is, and you should pay attention to. And it was very successful. A lot of people subscribe by email to that service. As soon as they read a news, they hear a news on the television, portal group used to produce. No, we don't have resources to keep doing that. That's one of the challenges with all of this. This requires a lot of intensive resources to keep up. And we haven't really figured that part out as yet. How do you sort of keep up with this misinformation? Because their machinery is much bigger than the machinery we will ever have in relation to that.
Yes.
Hi, thank you. Those were really, really fantastic talks, and I learned a lot. So I have a question about long COVID and vaccines. And so what do we know, if you could comment on what do we know about vaccine efficacy for preventing long-term symptoms of COVID? And there's obviously, I always get, every time I talk about long COVID, I get a question. If you get a vaccine when you have symptoms of long COVID, does that help you recover in any way? So I'd love to hear your thoughts on that from anyone.
Well, I mean, I can start off. I haven't done a search on that specifically, but I can tell you there's a lot of questions that remain unanswered about long COVID, right? So there are cohort studies right now that they're showing emerging data about risk factors for long COVID, but there are discrepancies, and the definition of long COVID is also discrepant. There are various definitions, and so there hasn't been the definitive study, in terms of a study that's looked at all aspects, immunological, looked at all sorts of, using standardized measurement tools to follow individuals. And if you're talking about a vaccine efficacy, we know the vaccine efficacy in terms of prevention of COVID, and we know the vaccine efficacy of even if there's a breakthrough infection that your complications are going to, but there are clearly no randomized controlled trials. So I would say the jury is still out on that, but I would say the best evidence, of course, is getting a vaccine, because it can prevent you from getting COVID, even if it's Omicron, and it can potentially prevent long COVID.
Yeah, and I think, so to follow up on Mark's point, I completely agree the quality of studies isn't there yet. There's some early emerging data that there is a risk reduction in humans, but it's modest. And to Mark's, I think, really salient point, one of the major issues with these trials going forward is the definition of what long COVID is, because it is almost certainly not one disease, right? It's a multi-organ disease, and expectations about which sequelae vaccination may prevent is likely very dependent on the biological basis of what symptoms, of which there are over 200 of long COVID, you're particularly manifesting. So are they vascular? Are they neurological, et cetera? The question about whether a vaccine helps people who already have long COVID is also being intensely studied. Most of the early signs are in animal models, and it's not clear how much they'll translate to humans yet, but there's been anecdotal suggestion it may, but nothing conclusive.
Great, thank you.
Thank you. I just looked at the time, so I know we're very close to lunch, but again, I just want to thank all of you for the work that you've done. And your teams, because I know behind you there's lots of people, and behind them there's lots of people. So thank you so much for using your talents so wisely during this pandemic on behalf of the public. So my question is to Mark. I don't know how you stay so calm, actually, knowing that 6 years ago you were already talking about some of the ways we should be doing this. And it's more of a question about community engagement, I guess, and thinking about whether you've taken a patient partner with you to these meetings, or who's been doing the speaking, I guess, to policymakers. Have you thought about ways that we can message things? Because I'm not sure, because of the way, we need different messengers is what I'm thinking about. And I just wondered if you had that experience and whether that had also made a difference.
No, it's me directly communicating with the bureaucracy and saying, it's a great idea, we're really supportive. But then them coming back and saying, oh, no, we just can't do it.
I'm just trying to think about that.
Political,
yeah,
actually.
So we've talked a lot about community engagement and just think about who's the messenger and who's and it's not that we're doing the messaging for people, but this idea of who's standing beside us when we're messaging.
It's a really good point, because I haven't had the time. I just
of
try
course.
every year. But it is a good point to use a different approach. I think it's a very good point.
Yeah. Great,
and
thank you.
I can say that he's not calm.
Okay, good.
He's calm here, but behind the scenes.
Okay, good to know.
He's deeply passionate about it. Any other specific questions? If I may permit to, are you wrapping up, or can I?
I was going to ask a question.
Okay, you go ahead then. I was going to.
Leyla, question for you. Point of care devices come all the time. And they're actually in clinical settings. There are very handful of them that really make it. As part of the data that you are presenting, which would be important when we are thinking about our CFREF application, how much of clinical validation of these point of care devices happen, right? A lot of them are small studies, you do little sensitivity and specificity, shows up really good, but when you take it to a clinical validation study or you set it up in RCT, they don't turn out to be that good. So what's the plan related to that, and does that fit in the context of what we are doing around the CFREF applications? That's the next stage of many of these devices. Any comment? And then I have a question for Amy, which might be a completely naive question, but I want to ask.
Yeah, so I think the challenge is that a lot of the what we call preclinical data, so it's more evaluation data, is done with prototypes. So the things we build in the lab, and then we have 60 to 100 or even less clinical samples, and when we do the evaluation. But for these devices to be commercialized, then the prototype has to become a manufactured prototype, and then the clinical study, a full clinical study has to be repeated. And then, so it's not, the published studies for these prototypes are not the final studies that will be done with the actual device. And so a lot of redesigns happen, and then those results might be different. So that's one of the challenges. So how do you do that with as much as a final prototype as possible that you can develop in a lab as something that's going to be manufactured? So we've tried to really take an already commercial reader, an already commercial chip, to make that as close as possible to the final prototype. But that's a challenge with diagnostic field and also therapeutics, and that's why a lot of people kind of run out of money in the process, right? So yeah, you identify the challenge that exists for sure.
Amy, this is totally, I understood only 10% of your talk because that's not something I work in. But from an aging perspective, we have not had very good success with animal models of aging, specifically around specific diseases. When you create these humanized mice models and you're testing different things in them, but they don't mimic the aging process per se. So how does this relate, and what do we need to do to move to that stage of innovation to be able to look at humanized mice models with aging mice models? Because we've had a dismal track record in that in many ways.
Yes, it is definitely a challenge, and of course I didn't say I could age them here, and there's reasons for that. So we can do aging mouse models in normal mice, right? Don Bowdish has an aging mouse colony, and we can perform vaccine studies in those mice, but again, they're not the human cells responding to the human virus in many cases, or bacteria. But in terms of humanized mice, the problem is they don't have the longest longevity. And actually, like in mouse models, you need to wait like years for them to be aged. And that's not possible with the humanized mouse models. So I guess I'd look at it more that we use them to make the most efficacious, and test the different methods that we know may be efficacious in older people prior to, so you get the best vaccine you can prior to going again into human studies where we would have to do the final tests in the aged population. Yeah.
Okay. Any other questions? I'm going to take the chair's prerogative and ask one last question, if I may, or maybe it's more of a comment to the question. Everyone hates that. And that gets back to Parminder's question to Leyla, actually. So I don't know about you, but before I came here, I took one of these rapid tests. They're available like crazy. I wanted to make sure that everything was cool. I was going to see all of you. And we are so used to this. We hadn't been in the past. Everyone's got a phone, even in the remotest communities, at least in North America, but perhaps even in low middle income countries, people have phones. They might not have the internet, but they have phones. So it seems to me that the kinds of platforms that you're developing, and if you link them to continuous monitoring devices to monitor the diseases, for example, that Mark had just outlaid about the challenges of the outlay, if I'm thinking, my phone has changed so much in the last 10 years. If I'm thinking 10 years ahead about this technology, now that we're all used to rapid tests and all this kind of stuff, and you're developing this technology, don't you think that the future is all about diagnostics, molecular rapid diagnostics, so that before you send your kids to daycare, that you can actually show them that they're fine, and just because they have the sniffles, this doesn't mean anything?
Right. Yeah, I think there's great potential. I mean, you talked about phones, and it's possible to even put these readout technologies on the processor on the phone, so you don't even have to have a dongle, right? The same way as we used to have wireless adapters and now it's all on chip, or cameras, right? So I think it would, it would be great if the field goes in that direction, that all these things are built in, and all you need to do is develop these cartridges for many different diseases. And
so we should make that a part of our pillars, but we have to make sure that it's done in an ethical way to protect our privacy, so that Bill Gates doesn't put another chip in a vaccine. Any other comments before we shut it down? And then I turn it over to Ina. Okay. Okay, well, thank you so much for this.